Hello everyone, I am Nikhil Seshagiri Rao. I am a PhD student from the Division of Applied Microbiology at Lund University. I am here to talk about my poster, Flow cytometric analysis reveals culture condition dependent variations in phenotypic heterogeneity of Limosilactobacillus reuteri. When we look at how the probiotics are produced industrially, first is the cultivation step where the biomass is generated. Then it is formulated using a lyoprotectant in order to keep the cells protected from the harsh conditions that it will go through in the next step, that is freeze drying. While these processes are going on, the cell needs to overcome a lot of stress factors. During the cultivation step, the process parameters pH, oxygen and temperature can act as stress factors. During formulation, non-compatible solutes can result in osmotic stress. During freeze drying, loss of water and low temperature can act as dehydration and freeze stress. As I am more focused on the cultivation step, I chose pH, temperature and oxygen to start with. With that said, the aim of my project is to understand the cell responses to critical process parameters during production that have a significant impact on stability of freeze-dried Limosilactobacillus reuteri strains. When I say cell responses, it refers to cell growth and morphology. Critical process parameters refers to pH, temperature and oxygen, and stability here refers to freeze-thawing and freeze-drying tolerance. The two microscopic images are the cultures of Limosilactobacillus reuteri but grown at different environmental conditions. This pleomorphic behavior has been observed before and might be an environmental stress response when exposed to a certain condition that has not been studied extensively. In this project, I wanted to systematically study the influence of process variables on cell morphology and correlate it to growth and freeze-thaw and freeze-drying survivability. I utilized the flow cytometry approach to determine correlation between culture conditions and growth, morphology, and survivability. All experiments were executed using the box penguin design with three variables, that is temperature, pH, and different levels of aerations, and three levels. This generated 15 experiments. These were cultivated for 24 hours and harvested by centrifugation. This was then formulated and frozen at 80 degrees centigrade. After 7 days, the samples were thawed and checked for morphology and survivability. Few of the interesting conditions were repeated to study the freeze-drying stress tolerance. To understand the effect of process parameters on cell growth and viability, a graph of percentage viable cells at harvest and total cells were plotted. As expected, a large variation in growth behavior and viability was observed between different conditions. A considerable number of cultures did not grow, although this was not correlated to poor viability. It was evident that temperature and oxygen had a great impact on the growth. This is a surface plot with temperature on x-axis and oxygen transfer rate on the y-axis. Blue color indicates low cell count and yellow indicates high cell count. This graph highlights that the growth was better at 37 degrees centigrade and 44 degrees centigrade with no aeration. Further analysis was carried out to understand the morphology using flow cytometer. As the cells pass through the laser, there are different detectors to capture the signals. The scattering profiles, forward scatter, provides the size of the cell, whereas the side scatter, depending on the cell size, provides the granularity and morphology of the cell. Another descriptor that we thought was worth looking at was the width. Pulse width is the time taken by the cell to pass through the laser. This gives us information about if it is a single cell or a doublet or more. In our case, it can be used to discriminate the longer cells from the shorter ones. The correlation between growth and flow cytometry morphology descriptors was made. This graph shows two flow cytometry morphology descriptors, forward scatter and side scatter height, versus growth, that is total cells at harvest. The results suggest that the flow cytometry based descriptors were able to capture subtle differences amongst the cultures. Large means of both the descriptors did not coincide with high cell counts under any conditions evaluated, which suggests that a high frequency of their occurrence is a sign of poor growth. To understand the morphological heterogeneity, I used pulse width distribution to generate subpopulations. The gates were created by using the create gates on peaks function on Flojo software. This generated 5 gates and I applied the same gates on all the 15 conditions. To check whether there is a correlation between the cells in a particular gate and growth, a plot of frequency of each gate and total cells were plotted. It was found that gate 2 had really interesting results. It can be said that the cells having good growth had over 60% of total cells in gate 2 when compared to the ones which did not grow had less than 45% of their population in gate number 2. This graph also gives information on the cell size. The ones which have grown well seem to be in gate 2 and seem to be smaller than the ones which have a wide spread, suggesting they were much longer. To check which conditions resulted in larger or long cells, surface plot with frequency of cells in gate 3, 4 and 5 combined were made with different process variables. From these graphs, it suggested that high pH, high oxygen levels and low temperature resulted in larger cells, possibly acting as triggers for chain formation, which was inversely correlated to cell growth. This can be backed up by the pictures taken using the light microscopy and scanning electron microscopy. Cells cultivated at 30 were longer than the ones cultivated at 37 degrees centigrade and 44 degrees centigrade. If the scanning electron microscopy pictures are examined carefully, one can observe that some of the cells cultivated at 30 have not divided completely. This might be one reason why we see them as one long cell in light microscopy, whereas it can be two or more joined together without division. To investigate the differences in freeze-thaw stress tolerance between the cultures, cells were subjected to one cycle of controlled freezing and thawing, and the survivability was determined. A large variation in survivability was observed. There was no clear correlation to cell growth or any of the morphological descriptors. This gives us 4 clusters. The one we desire our cells to be at with good growth and tolerant to freeze thawing. The second one, growing but then poor freeze thawing tolerance. The third one, poor growth but tolerant to freeze thawing. And the fourth one, poor growth and has poor tolerance. The impact of freeze-drying stress on bacterial cells was studied on the cultivations that displayed variation in cell distribution and at the same time had good growth. Freeze-drying survivability was higher for the cells cultivated at 37 degrees centigrade and 30 degrees centigrade than for the cells grown at 44 degrees centigrade. Subpopulation distribution was examined for 3 conditions at harvest and after freeze drying. For bacteria grown at 30 degrees centigrade, there was a relatively even distribution of viable cells in pulse width gates 2 to 4. This was different from bacteria grown at 37 degrees centigrade and 44 degrees centigrade, where majority of the cells were found in gate 2. Also, freeze-drying treatment did not change the pulse width distribution to a significant degree, which indicates that stress tolerance was equal for bacteria of different sizes. To summarize the whole project, we now have a flow cytometry pipeline which could be used for analyzing and correlating between environmental factors, and cell morphology of Lactobacillus reuteri during cultivation and subsequent freeze-drying processing. The pulse width distribution parameter can be used as a process analytical tool in process control of morphology during fermentation. It is industrially relevant as it can be used to check the robustness of cells after a certain stress. We also saw that there was poor correlation between microscopic analysis and flow cytometry analysis for morphology, which suggests that the flow cytometry analysis gives out more information than not just the cell size. Suboptimal conditions such as high pH, high oxygen levels, and low temperature resulted in larger cells, which was inversely correlated to cell growth. And finally, a high robustness of reuteri towards freeze-drying stress was observed for cells cultivated at growth optima, and this coincided with good growth and a cell population consisting of smaller cells and a narrow size distribution. I would like to thank you all for listening. You could connect with me on the email ID that has been provided below, or you could scan the QR code to reach my LinkedIn account. Thank you.